Hello and welcome. My name is Esiason Rodriguez, and this is my colleague Sameerah Mohammed, and our project is to explore salinity tolerance in pistachio trees by measuring stress responses at the biochemical level.
California has experienced several droughts throughout the last decade, specifically the 5-year drought from 2012 to 2016. These recurring droughts have caused California's agricultural industry to suffer due to the scarcity and cost of clean water. Our research aims to analyze the pistachio tree's unique biological response to saline irrigation and means to develop a better understanding on why some plants are more environmentally tolerable than others. Previous studies have shown that pistachio trees are highly tolerable to stressful environmental conditions such as saline irrigation. This tolerance is due to the biological mechanisms which produce life-sustaining biochemicals. Since these biochemicals are produced in response to stress, we can use them as markers to show how a plant responds to its environment. For this reason, these chemicals are referred to as biomarkers, which we will take a look at in the next few slides. We aim to analyze 3 different variables. These variables were rootstock, treatment, and leaf age. Rootstock allowed us to see which of the 2 types of pistachio species had greater stress response, UCB1 or PG1. Treatment allowed us to see how saline irrigation influences the production of biomarkers, and leaf age allowed us to see whether younger or older leaves had a greater stress response. To analyze the pistachio leaves, we first had to gather the samples from a lysimeter field overseen by Dr. Banuelos from the USDA Agricultural Research Service. Once these samples were shipped to our lab, we then performed ethanolic extracts, which were used in further analysis by use of various biochemical assays.
Here are some pictures of pistachios and the farms in which they are grown. To the left, you can see how our samples were grown at a USDA research site in flower pot-like apparatuses called lysimeters, which allow Dr. Banuelos to mediate individual growing conditions, such as what type of soil and what water treatment they receive. As discussed earlier, biomarkers are chemicals which vary in concentration depending on environmental stress conditions. The biomarkers that we analyzed were photopigments such as chlorophyll, the osmoregulator proline, and phenolic compounds which serve as plant antioxidants. Here are the 3 photopigments that were quantified. Each pigment serves its own unique role in photosynthesis. Chlorophyll A is necessary for photosynthesis, whereas chlorophyll B and carotenoids are accessory pigments that allow for greater amounts of wavelengths to be utilized when needed. As you can see, there are different absorption patterns for each photopigment, which is dictated on the graph to the right. To measure photopigment content, we used a nanodrop 2000C, which is able to quantify a sample's light absorbance.
Proline is known as an osmoregulator in plant species. Osmoregulators mediate the water pressure inside the plant cells, and water is used to carry vital nutrients through a plant's vascular system, so if the osmotic pressure is reduced, certain nutrients may not be efficiently delivered and absorbed. The osmoregulator proline was quantified by reacting sample extracts with anhydrous reagent and glacial-acetic acid, which induced orange color formation, revealing the presence of proline. This orange color development was then quantified by means of UV-vis spectroscopy, which is able to quantify the concentration of proline based on the sample's light absorbance. Here is a picture of the UV-vis biotech plate reader and the picture of the sample plate used. As you can see, there is evident orange color formation in both sample pictures. The phenolic compounds in plants are known to act as antioxidants because these compounds are able to restrain production of reactive oxygen species. Phenolic compound determination was quantified by reacting leaf extracts with 10% Folin CaO2 and 700 millimolar sodium bicarbonate. This reaction produced a blue color due to the presence of phenolic compounds. This blue color formation was also quantified by UV-vis spectroscopy. In these samples, we see evident blue color formation.
Based on our photopigment analysis, we found that age showed the most significant effect, whereas treatment and rootstock showed no influence. Here are the 2 graphs that represent the photopigment content. On the y-axis is a photopigment concentration, and on the x-axis is a treatment concentration. In comparing these 2 graphs for PG1, we can see the young samples were significantly higher in concentrations of photopigments than the old samples, especially for chlorophyll A. Overall high sample variations, as expressed by the error bars, make any treatment dependence inconclusive. Here you can see our proline data. The young samples are represented by orange, and the old samples are represented by pink. In these two graphs, proline content is on the y-axis and treatment concentration is on the x-axis. As you can see for both graphs, young leaf samples had greater proline content than the older leaf samples. Neither the rootstock nor the treatment showed influence on the production of proline due to the high data variation represented in the large error bars seen here. As for the phenolic compound analysis, age and rootstock showed the most prominent influence on phenolic compound production, but treatment showed no direct influence on the amount of phenolic compounds produced. For these two graphs, phenolic content is on the y-axis and treatment concentration is on the x-axis. We can see here in both graphs that in general, the young samples are higher than the old samples, showing that the young leaves contain greater amounts of phenolic compounds. We can also see that the right graph, which is for the UCB1 rootstock, is much higher in overall concentrations than the left PG1 graph.
In conclusion, through a statistical analysis of variance, age showed to be the most influential variable when it comes to the production of photopigment, proline, and phenolic compounds. Therefore, consistent leaf sampling is imperative. Rootstock influenced phenolic compound production, but not photopigment or proline production. As for the treatment variable, there was no direct correlation between saline treatment and biomarker production. Other studies have shown that saline treatment greatly influences biomarker production, but current findings prove to be inconclusive. We may be able to accurately conclude our findings if we increase our sample size, as well as if we perform more replicate readings based on the samples we already have.
This in-depth understanding of a plant's biological response to stress can be used to develop more sustainable agricultural practices, such as breeding and selecting for environmentally tolerable plants so they can succeed in a given environment. One of these sustainable practices, which utilize a plant's ability to tolerate adverse conditions, is irrigating with saline water, which has shown to produce successful yields while reducing the cost of irrigating with clean water. This is just one example of how our understanding of a plant's biological response can be used to develop further sustainable agricultural practices.
Many thanks to Dr. Gary Banuelos from the USDA Agricultural Research Service and the CSU Agricultural Research Institute. We hope that you enjoyed our presentation. Thank you.